Welcome everyone. My name is Dev Chakraborty and I welcome on behalf of the IUFRO Working Party 2.5. And we have just started our new webinar series. And before I can introduce our speaker, let me just walk you through very shortly about IUFRO and our working party. So IUFRO is a non-profit, non-governmental international network of forest scientists, and it has a headquarters in Vienna. And it is one of the largest, or probably the largest, conglomeration of scientists, policymakers, and practitioners, anyone who is related to forests that we work together. We are very close, and every 5 years, the IUFRO also organizes the IUFRO World Conference, and it is really dedicated from a very, very long time on various issues related to forest and forest sciences. IUFRO have now 9 divisions, 13 task forces, 60 plus research groups, 180 and counting working parties, and 4 different special programs and projects. And among these 9 divisions, the division 2 focuses on physiology and genetics. And within this working party, within this division, we have the working party 2.05 that focuses on breeding and genetic resources of Pacific Northwest conifers. So this Pacific Northwest is a very, very special area, very, very special part of the world where many different conifers and many different broadleaf as well. It's for forestry, it's a very, very important area. And this working party focuses on breeding and genetic resources of conifers that comes from this distribution range of North America, British Columbia, all the way to California and elsewhere. So what I wanted to stress here is elsewhere, because these conifers, for example, Douglas fir, Sitka spruce, Western hemlock, all these species are not only very, very important for Pacific Northwest, but also for Central Europe and many other parts of the world where they have been planted and for a very, very long time. And now, for example, where we are based in Austria, and also in the Central Europe in general, Douglas fir, Sitka spruce, and all these species are very, very important and has been discussed or are currently under discussion as an alternative for many of our native conifers, which are vulnerable under climate change. And my name is Dev Chakraborty. I coordinate this working party, and with me, I have my colleagues Marie Suntama from Skogforsk, Sweden, Marie Vance from the Ministry of Forest in BC, Canada, and Nick Ukrainets, also from Canada, from the University of British Columbia. So today we have just started with our series of webinars, and there can be no better guest or speaker than Professor Milan Lstibůrek, and he will talk today about assisted migration, pollen-based issues, rolling front landscape breeding, and forest adaptation under climate change. Professor Milan Lstibůrek is an internationally recognized figure in this topic, and he is especially well known and recognized worldwide for his work on breeding without breeding and rolling front landscape breeding. So without further delay, I would ask Milan to share his experiences and his knowledge with us. So I will now stop sharing. And Milan, thank you very much for your time. Please, the floor is all yours.
Okay, so let me see if it works.
Please.
Can you all see the screen?
Yes, we can hear you very clearly.
Excellent. So Dev, thank you very much for this invitation, and thank you all for joining us today. And I very much appreciate that you're spending your time with us on this subject. So first of all, I would like to say that I have I'm providing you a link where you can find the PDF version of this presentation, and you can also find all the papers that I will refer to. So this is an online repository, so you can access it and download all these files. Also, I have a few points that I wanted to discuss before I start with the actual presentation. So first of all, I try to keep updated information on my website, and I try to put things in a bit in a perspective, so you can read how the different papers relate to each other, how they relate to the current issues in forest genetics. And then I also have this GitHub page that I already shared, and that includes computer code for designing seed orchards and other things. So you may visit it, you may download it, you may use it. You can also contact me by email, and I'll be happy to answer any question, or we could set up an online meeting if you want like one-to-one discussion on any subject. And we also organize schools, summer schools. I teach 1 week long introduction to quantitative genetics, meant for international PhD students as well as senior scientists. Basically, whoever is interested in quantitative genetics can join. It's organized here in Prague at our faculty. You see the picture on the left, the upper left picture. We have a beautiful campus here, and so if you're interested, you can join. And we also provide credits for students. There is another event which is organized annually in Austria, and this is done by Dev and Silvio from BFW. And as far as I know, the next year it will be most likely organized in Vienna, and there you can learn about assisted migration, and you can try calculating all the aspects of that using actual data and so on. It's also a week-long course, and we can also provide credits for PhD students. And I sometimes organize workshops, such as how to design seed orchards, where people would bring their computers, bring their data sets, and I teach them how to do it, and either physically or online. So if you are interested, talk to me, and we could come up with something. And lastly, there is a link to my web page, to my WhatsApp group, and there I provide some information about my papers and anything that's new. So if you sign up for that, you will get that information from me periodically. Now, so let's get started. it's quite difficult to and challenging to come up with the right approach how to deal with how to manage forest gene resources under climate change. So all I could think of are these two concepts, and initially I thought of each of them independently, so they just occurred to me, but over the time I thought that it might be a good idea to present them together because they could be connected, and I think they are quite powerful when they are connected. So I will try to explain that today. So for most of you, this is obvious, and I just want to say it anyway. Forest trees, unlike the model organisms, are very complicated, and from a basic science perspective, but also from applied tree genetics and breeding perspective. Quite challenging organisms for scientific exploration and breeding progress. So I just go quickly through these complications. So they are long-lived organisms. They're large. They occupy space, a lot of space, and they live in heterogeneous conditions. Forests are used for many different purposes. So there is this multi-objective or multi-criterion optimization of the forest use, which complicates everything. And they're dominantly undomesticated species. From a genetics perspective, they're mostly outbred organisms, which is again a complicating factor. They're known for huge genome size. Most of the traits that are related to fitness are have complex genetic architecture, meaning they are influenced by many genes and most with relatively small effects. Often they are exposed at later life stages. They're also known for fast LD decay, low heritability traits, quite often significant genotype by environment interactions. And unlike the crop breeding or animal breeding, we really have to put a lot of weight on genetic diversity when we perform selection. So that makes the progress much slower. The new era of genetics brought us these pictures. So this is an epilepsy in humans, a typical complex genetic architecture where we see most of the trait variation being due to a large number of genes with small effects, and some genes that have higher effects, but they only explain a relatively small proportion of the overall variation that we see. Now, what happens is that we live under abrupt changes of climatic conditions. This is the, I would say, if not the latest, then rather new data from NASA. And you see the spiral graph of the global temperature, and I don't want to say more about it because it's self-explanatory. But what should bother us as forest geneticists is that it pretty much influences everything. So in the simplest equation that we all know from the basic textbooks, phenotypic value of a specific trait is composed of genetic and environmental components. And what the climate change does is people may think that it only affects the E, which is the environmental component of this equation, but it does not. So the climate change manifests itself through the evolutionary processes such as the natural selection, migration, and random genetic drift caused by fragmentation and other issues. And we all know from the basics of population genetics that these factors will manipulate allelic and genotypic frequencies. So they will alter the genetic composition of our populations. And when they do, and the allelic frequencies change, you will have changes in all of these variance terms and the interaction terms as well. One has to also see the epigenetic component and this nice expansion, which is basically just a long equation. You can add more lines to it, considering the epigenetic component as shown by Wang and others. And it's easy to see that the climate change will also impact these components. So what should bother us is that we can no longer predict the outcome of both the artificial and the natural selection. We cannot thus predict the outcome of breeding or conservation programs. So that makes it much more complicated, because before we used to think about the environment as pretty much constant. Even it was possible for it to vary, but we didn't think of it as a systematically changing factor. And that changes pretty much everything, also in terms of the management of the gene resources. I would like to go back now, and over 30 years ago, I got this nice work report from Skogforsk and from the Forestry Research Institute in Sweden. And already like 33 years ago, because this is a reprint, there was a discussion on climate change and how to adapt breeding into that scenario. So what you could see is the delineation of breeding zones in Sweden, and there is a brief but interesting discussion that the material could be moved along these main gradients and to face the issues of the climate change. So I think this is a very nice example that this is not a new topic, and this has been around for a long time and has been discussed. What I like about this paper is that breeders have thought that one can combine both the local adaptation and the assisted migration, or you could say movement of forest reproductive material. And I go back to that idea. I will return to that idea shortly. But before I do, I have to say a few words about how this whole thing started, and sort of provide you with the background of the rolling front landscape breeding. And I go back to this breeding without breeding concept. It's not the only concept that was around during that time. And this emerged as a consequence of the availability of DNA markers and the opportunity to reconstruct pedigrees relatively simply. So with just 10 to 15 highly polymorphic microsatellites, you can get a relatively accurate pedigree information. And that was a nice opportunity to explore some simplification of tree breeding. And so just to explain briefly, we start in generation N, which is on the bottom of the page, and then control crosses are made to produce progenies that are planted in the progeny trial. And then based on their screening and selection, new parents of the next breeding population are selected, and they form the second generation, and so on. This is called the recurrent selection, and it's been applied in forest tree breeding for decades, and it works. Okay. In every generation, top individuals are typically grafted in seed orchards and used to provide genetically superior forest reproductive material, typically seedlings that are then used to plant forest stands. So over the generations, you get the increased frequency of favorable alleles that are affecting the traits that you want to improve. And as such, we said, well, what about if we let the nature do the pollination? So assuming open pollinated species, we said, well, let's just collect a bulk seed and forget about the trials. So we could just collect bulk seed, establish forest stands, and then use the DNA markers to reconstruct the pedigree, use advanced technology to provide phenotypic records, and then select the material as the parents of the next generation. So here you retain the necessary component of tree breeding of outbred organism, that is the genetic recombination, testing and selection. So all of it is retained in this concept, but you don't have to do a single control cross, and you don't have to establish these nicely groomed progeny trials, because you can rely on forest stands that I will show you that shortly. So some people were quite pessimistic about this, and we are quite conservative in forest tree breeding. So I will show you what we did in Austria. And this was a project that was financed partially by the forest owners and by the Austrian Research Promotion Agency. and it was managed by Thomas Geburek and Silvio Schuller from PFW. And the assignment was simple. So these are the first generation seed orchards of European larch, and they're too old, and they wanted to move to the second generation seed orchards, but there were no progeny trials whatsoever. So what we did, we found multiple forest stands that originated from these first generation seed orchards. There were actually 21 of such stands along the major environmental gradient, which is the altitude. And there were two crews that measured the trees and collected DNA samples. But I have to explain that the DNA collection was not performed from all the individuals, because by that time we already conducted some research on simplifying the protocol, and we knew already that we don't have to genotype everything. So I will explain that a bit later. So this is the result. On the left side, you see complete pedigree that we discovered. So in total, this is 1,024 offspring that were genotyped out of the 4,000 and so trees across from the 21 stands across 4 climatic regions. And based on the markers, we reconstructed the pedigree. So we also genotyped the parents in the orchard, and so the and then use the multi-trait, multi-site genetic analysis in ASREML. And I asked Professor Gary Hodge from NC State to supervise the genetic evaluation, as he is really renowned for such expertise. So I wanted to make sure that it's done the super way, and we found very good results. So the heritability for height was 0.23, and with the error 0.07, and the wood density had a heritability 0.3 with the error of 0.07, and this is similar to conventional trials. So this is a proof of concept, and it was published 5 years ago. And I just want to say that BWB works. And there is a series of new seed orchards, second generation seed orchards now in Austria based on this. And this is the timeline. So you see that we have started in 2014, and already in 2019 we were establishing the new seed orchards. So without a single cross, without a single progeny trial. So this gave me some hope to continue, and I thought, well, there is a lot of things that are still missing in this concept. Now that we have to worry about climate change. So I knew from my studies at NC State there were interesting ideas and suggestions, and one of them earlier, and one of them was a rolling front strategy. And you can find these two papers that are extremely interesting. I encourage you to read them, and they provide the basics of what I suggest, except that I combine it with the BWB component. So I will explain that simply without equations. So let's say we have a seed orchard, which we may call a breeding arboretum, and we start establishing forest stands from this seed orchard. So let's say in 2000, we would establish, using a bulk seed, conventional forest stand from this seed orchard. Typically, you would establish a number of such stands in different locations. So I will call these locations sites. So here we have a site 1, and I'm establishing a forest, one forest stand in year 2000. Okay. In 2 years, I will establish another forest stand in the same location, and I will also establish it in other sites. And I will keep adding more such stands. They will be even aged. They will be single species to facilitate genetic evaluation. Okay. What happens here is that these stands will grow and eventually reach a selection age. And just for the simplicity, let's assume that the selection age in this example is 12 years. Publish these stands every second year. We could do it every year, or we could do it every third year, or every year that we have sufficient amount of cones in the orchard. So it may vary by species, but you see that we have this emergence of selection candidates across all sites. They're shown as the red line. So these stands, this is a mean height. This is just simulated in MATLAB, and it's not based on real data. And you see that there is a growth in each stand. So 12 years following the establishment, I will start having selection candidates from this in this first stand. In 14 years, I will have already 2 stands I can choose from, and so on and so forth. So I will keep adding more and more selection candidates to my candidate population. What's really important is that I expose each stand, which is a single recombination event, to a unique set of climatic conditions. So the specific life stages are exposed to unique conditions. And I consider this to be very important under climate change, as we need to take care of or keep track of the dynamics of the change that we cannot really predict, but we can evaluate it periodically as we are adding more and more individuals, and we keep phenotyping everything. So all stands originate from the same parental source. So there is no shallow pedigree. This is really stronger genetic relationships, which is good to create the covariance that we need for genetic evaluation and selection, and we utilize the multi-site, multi-age genetic evaluation model. And then what I've seen, what I have showed you are these blue lines. So you're creating these forest stands at each site, and eventually you will start with your genetic evaluation, and you will be bringing the best genotypes to what I call a reservoir, and that's where you prepare your grafts for the seed orchard. So you have a core subset, which is a reproductive part of the orchard where you collect the seed as normal. And then there is a reservoir where you are moving the top candidates to eventually become parents, and that could be then moved to the core subset and become the parents. And so if you keep doing this for a long time, then the breeding arboretum is sort of is staying there indefinitely. There are no breeding cycles. You just keep shuffling the genotypes in and out based on the periodic evaluation across all the sites. You will also keep establishing these sites, so it's an ongoing process. So you have the selection candidates. So the superior individuals in forest stands available for possible inclusion in the arboretum, and they can be moved to the reservoir. I'm not going through the optimization. You can find it in the paper where I propose how to use mathematical optimization. And that optimization is done and every time you measure trees again and you add new candidates, you re-optimize everything, and the decision is based on the data. It's not based on your decision, it's based on the data. If there is a better candidate and it makes sense at that moment to move that candidate to the reservoir, it's done. And over the time, some of the candidates from the reservoir could be removed, but some would be moved to the core subset. Now, this process could be extremely either bypassed or simplified by using the top grafting, something that's been mastered by NC State breeders, particularly Steve McKean. He's done a great job on this, and I encourage you to read that because the top grafting is really superior. So, and it would fit well in here. So you will bring the science of the top genotypes, and you can basically graft them on existing trees in the orchard. And if you combine that with the control mass pollination, it's becoming a super powerful strategy. I'll talk about the control mass pollination when I explain the pollen-based assisted migration. And I promise you will be impressed. Some of you may know, but you will be impressed how flexible it is, and that it's really used it's used a lot, and it could be put into practice relatively simply. And eventually, genotypes are removed from the core and being replaced by better candidates. And this is how I like to see seed orchards. So a simple way to move the trees. It may not be realistic in all orchards, but I think this is a nice example. And I promised you to say a few words about the genotyping effort. So you don't have to genotype everything across these stands. In fact, we showed before that only a relatively small fraction of trees needs to be genotyped. And there are, I think, there are two papers that I would recommend you to read, and I think they are on the in the repository. One of them is the paper on the right in DGG 2011. On the left, you can see a paper that may surprise you as we show that you don't actually need to use random samples to estimate genetic variants, which sort of violates the regression ANOVA type of genetic evaluation procedure. So what we show is that you could use only the top phenotypic proportion of the population to get an unbiased estimate of the neuroscience heritability, which you then use in the blob predictions. So this approach may significantly reduce the cost by eliminating the random population subset, which we assumed both initially with Yusri Al-Kasabi and also in Austria. Now, I would like to explain this a little bit. So, this was one of the criticism of the breeding without breeding, that if you get a pollen contamination in seed orchards, and so it wouldn't work. Well, I claim that since you are focusing only on the top phenotypes in the offspring, then if you have poor parents, genetically poor parents, they are shown as red. So on average, you will have a poor parent sending its pollen, which means the haplotypes, to the mother in the orchard. Then their offspring would on average be lower ranking compared to an offspring of two parents that are in the orchard and that are genetically improved or superior. And I showed this in a paper in 2012, and we had two, I believe, two empirical studies verifying this. And so this has really bothered me. Like I thought, well, this is a nice set of equations, but I thought, well, what about we change the approach that the input becomes the output, and suddenly this emerged, which is, I thought, well, what about if we focus just on the right truncated proportion of the distribution in the parental and the offspring population. So let's say you would only look at or identify trees that are the tallest among the parents and the tallest among the offspring, and you forget about everything else. And could you estimate heritability? And it turns out yes, and we published this paper in 2018, and so the conclusion is that under the assumption of Hardy-Weinberg equilibrium, the heritability is directly related to the likelihood of a phenotypic subset of parents passing their respective alleles onto the corresponding phenotypic subset of offspring, which means that you would be measuring the top or genotyping only the top ranking individuals of the population, and only the and genotyping on only the top ranking individuals in the parental population. And you will be asking a question, okay, is this offspring originating from parents from the top phenotypic segment or not? And you go through these offspring above the truncation, and you get a proportion of those that are actually originating from that subset, and then you provide the phenotypic variance of the parental and offspring generation, which you can assess, and then you plug it in to the equation that we provide, and that provides you with the unbiased estimate of the heritability. So I like to think about this as a new definition, and it sort of opened my eyes on what the heritability actually means. So it's another way to look at the heritability. But for us, it's also interesting that we can now only focus on the top phenotypic segment of the populations, as I already described. And if you're interested in quantitative genetics, we use the data of Francis Galton and prove that we get the same results as he for the human height. So I think I'm going to conclude on this. So first of all, the traditional tree breeding. It has discrete generations and is based on the assumption of a static environment. So the progeny trials are established once per cycle. So a relatively narrow environmental window, but a selection decision is made decades later, implicitly assuming the G/E effects are stationary across time. And because the climate is shifting, each discrete cohort only reflects a snapshot of past conditions. And when the breeding values are estimated with that static cohort, the estimates would be biased towards adaptation to an environment that may never recur. But the concept that I propose is that instead of one large recombination event in 10 to 30 years, we have recurrent recombinations every, let's say, second year. And each cohort is exposed to unique environmental conditions, which is very important. So all the life stages. And the simultaneous genetic evaluation is conducted across all sites and ages under a mixed model framework. And if one cohort encounters a severe drought or frost event, the response of its families contribute information to the breeding value estimation across the entire population, and this allows capturing adaptation signals to extreme or novel climates. That could be combined with what we propose as a pollen-based assisted migration. I won't talk too much about this. Think about the assisted migration, the conventional assisted migration. But let's think about seed orchards. There are a lot of seed orchards across Europe and elsewhere. This is just one database, so excluding data from other countries, but you see we have the opportunity to collect pollen from source seed orchards and use that pollen in target seed orchards based on the assisted migration methodology that already exists. So instead of moving seed or seedlings, we move pollen and hybridize with the pollen in the target location. So this is a paper that you will also find in the repository. And it's a work of my PhD student and our team, a larger international team that we put together. This is an example of the control mass pollination that could be used for this purpose. But as I said earlier, it would be extremely useful and efficient in the context of the rolling front landscape breeding that I described before, and would make it super efficient. Now, you may be aware, some of you may be aware, that 2 million seedlings annually are produced through the control mass pollination. I hope, David, this is correct. Is it 2 million or is it 20? I think it's 2 million. Correct me if I'm wrong. But it's about 1/5, 20% of the seedling production in loblolly pine in the southeastern United States. And you could say, well, it's an intensive program, and you know they have money and for that. And I would say, well, we have to start thinking differently. It's going to be more and more expensive to keep the forest alive, and we have to start thinking about forestry differently. We will have to start putting more money into forest than we were used to, just to keep the forest alive. So this is definitely worth the investment. You could also see the supplemental mass pollination on pictures D&E, but I won't discuss it. You can read more about it in the papers. There are current problems of the assisted migration solution that we offer. As I said, the hybrid progeny offers some benefits, and what we really like about the hybrid approach is the epigenetic priming, meaning the pollination and the fertilization and the embryogenic development takes place in the target location where you're gonna use and plant that material, and that's super important under climate change. It's important generally, but it's super important under climate change. There are also phytosanitary risks. And I also forgot to say there is, of course, the component of local adaptation. And when you have tree breeding locally, you don't want to just replace populations, as you will find superior individuals within your program. So this is more like an enrichment strategy or infusion of the alleles that are suitable. Phytosanitary risk. Pollen is considered basically risk-free regarding the pathogen transmission. That's not the case with seeds or seedlings. And there are struggles. Stakeholders, nurseries don't like if their business is jeopardized and things like that. So all of that you could read. We've also conducted this computer simulation that hasn't been done before. If you don't include the additive genetic component, you're basically ignoring the fundamental theorem of natural selection. So you have to add together additive, dominant, epistatic components, allowing you to consider both the direct genetic effects as well as, for example, outbreeding depression, heterosis, and factors like that. So we show how to do this type of modeling. And this would be what I consider the merge between the rolling front landscape breeding and the pollen-based assisted migration. So you have a Vienna forest, you have a seed orchard or a breeding arboretum with the sides, and this would be a rolling front setup, and you could have a different rolling front setup somewhere else, and then there would be a pollen-based assisted migration in between these. So you have the element of local adaptation and you have the element of assisted migration in it. Now, some of you may ask the question also, what about the genomic selection? And I would suggest the following. So if you want to move extremely quickly and you believe there is no climate change or there is no environmental change, or you could or you work with short rotation species, but by short rotation I mean like 3-4 years, because you see from the spiral graph things are moving quickly. So then you could lean to genomic selection, but keep the multi-environment training updated, frequently updated. If you, on the other hand, believe that climate is changing as we see on the spiral graph, and particularly work with long rotation species, and acknowledge the fast LD decay, then I strongly suggest you to use something that I have suggested, or its alternative, and supported by genomics. So what I suggest is compatible with S&P genotypes. So you use it to estimate relatedness. So you work with the G matrix, the variance decomposition, diversity control, early culling, etc. All of it is compatible with what I said. We have or my two PhD students will have a presentation in Spain next month, and I encourage you to listen to Christy, who will talk about the impact of errors in marker data on the G matrix, and particularly on the narrow sense heritability. We have a paper in, I hope the paper will be out before we go to that meeting. And I was literally shocked by that. So I hope you will enjoy reading this paper. David will provide more in-depth introduction to pollen-based assisted migration in Madrid. Okay, and with that, I'm sorry it took me longer, Dev, but I guess I'm still on time, and I could offer you to continue with the discussion if you're still alive, or we could set up individual discussion anytime you prefer. So please let me know. And again, thank you all for listening to me for 1 hour. I hope I haven't disappointed you.
Thank you, Milan. Thanks for the great talk. So before we have a few minutes left for to take some questions, there is a question in the chat by Berika Beritze. So she writes, She will check the articles, of course, but her concern is that, can you please elaborate on genotyping? As I understand that you only verify that strong grown trees are from strong parents, and then cycle goes on.
Well, let me see if I understood. So, assuming the paper in genetics, where we look at the tallest parents and the tallest offspring. So, let's see if I could. Let's see if I understood correctly. You can explain it. Let me see if I could. I don't see I can. Okay. All right. So you know, it doesn't matter what type of markers you use. What you need is to have reliable pedigree information. So you need to know whether this parent is my dad and whether this parent is my mom. And so for that purpose, you could either use highly polymorphic SSR markers, and you would need, I don't know, 15 of such markers give you sufficient statistical power already, or you could use much larger set of SNP markers that will do the trick as well. Okay, so it's just that a pedigree assembly that you need. And then you're asking the question, is this my dad or not? If I am above the truncation, is my dad above the truncation, and is my mom above the truncation or not? And so I'm just counting these occurrences, and then at the end I plug in this information to the equation, and I get the heritability. I don't know whether that answers your question, but
we don't have much time. There is a hand raised by Greg O'Neill. So I'm specifically allowing him, since if I am not mistaken, it's Greg from Canada, and I'm specifically allowing him to speak up. I have unmuted you, Greg. So if you have a question, please go ahead.
Thank you. I appreciate that. Good morning from British Columbia. Thank you for the presentation, Milan. Milan, I have a question, a fundamental question about how pollen-assisted migration might be applied in Europe or anywhere else. For example, in conventional assisted migration, if one is planting at a site with a mean annual temperature of, say, 10 degrees, similar to Vienna, one would get seed from a location maybe 3 degrees warmer, say 13 degrees, and then use that seed to reforest a site near Vienna to account for past and future climate change. However, in pollen-assisted migration, one would need to go to a location of 16 degrees to get pollen if you're applying it back to the Vienna seed orchard in order to get seed that's adapted to 13 degrees. So is it realistic to look for pollen in a location that's 6 degrees mean annual temperature warmer than your planting location, given that the entire range of mean annual temperatures across Europe is almost 6 degrees.
Yeah, I got it, and thanks for that question, which is excellent. Actually, that's based on the assumption of the additivity, right? So that you have a half and half. But based on the simulation work we've done, we don't necessarily think that's the case. And actually, what happens is that you have factors such as the epigenetics that I described, and that contribute more. So likely contributes more. So so of course this has to be done on a species specific basis. And I promise you that with David, we are now focusing on research where we are tackling with some of these questions. Because I understand where you're going from, but what I could say immediately is that this is not an additive scenario, and most likely you won't need to go that far. And we like to see also about, and this is how we present it. We like to see about, we also like to retain some of the local adaptation. And that is, the climate will keep changing, right? And so you cannot replace all the forest with something else. And you cannot replace it at the same time. And so some flexibility there, and something's telling me, well, we should retain as much as we can locally, and then refresh it with haplotypes that are from basically distant haplotypes, but not necessarily twice as far as using conventional assisted migration. And I promise we have an ongoing research, and I think we will present something that's new and that may change our view on how the assisted migration might work in this context.
Thank you. I look forward to hearing more about that. Thank you, Milan.
Yeah, you have the same question that we had. And actually I was quite new to this, and it surprised me that there was not more on genetics in the assisted migration in trees. There was a little work done on in computer modeling, and I see a lot of questions myself that I would like to have answered before making realistic proposals to foresters, tree breeders, because they keep asking questions, how can I implement it, assisted migration? I have a breeding program, what should I do, and things like that. And we don't have all the answers now. We are behind. So
well,
thanks, Milan, and
best of luck.
Thanks, Greg.
Thank you.
Thanks for actually also commenting on David's work. It was really nice to hear perspectives from you guys. It's really great. Although we are running out of time, we have actually surpassed our given time. I will just take one question that was posted by Laura in the Q&A. She writes, I was wondering how would the rolling front affect the genetic diversity of the breeding arboretum? Wouldn't the consecutive selection on top parents may end up in related trees in the parental cohort?
Yeah, well, eventually it would. So when you look at the older papers and dissertations back in, I don't know, while I was still a student, so there were different ideas. So sublining was a great idea that Steve McKean published in Silva Genetica a long time ago. And there was, of course, the the infusion of the unrelated material from either wild stand or from neighboring breeding zones, etc. So we have now the luxury of using the pollen-based assisted migration as an enrichment option in a long run. So from a short-term perspective, I would say 3 to 4 generations is not a problem. But again, in terms of the generations, I should not actually call it generations, right? So, but I mean, the time needed to accumulate the relatedness using this concept, depending also on how big the populations are kept and so on, so the resources available. But this pollen-based assisted migration is actually useful, helpful, because it's an enrichment. So you are effectively increasing the effective population size of your population, and it's done just faster than the nature would do it. You know that the nature is using migration to increase the heterozygosity. Well, if we use the pollen-based assisted migration, we just do it faster.
Great. Thank you, Milan. Milan, we cannot hear you properly now.
Okay, should I repeat it again?
No, now it's fine. Actually, we cannot take any more questions, but I promise you there are some more questions from Ram Gopal. He asked whether this study is applicable to all trees or conifers. Ram, I think you mean pollen-based. So, Milan, can you answer in yes or no? It can be applied.
No. And what I would like to do is just initiate a discussion, scientific discussion. And I'm not necessarily right here. It's just my reasoning, and it's not applicable to all species. So you just take it as an option and think about how to adapt it to your species. And if you want, we can discuss it together. Feel free to contact me.
Yes. Thank you everyone for joining us. David, Matty, you have questions, and there is one more as well, but we don't have time yet. I will make sure that I take your questions to Milan, and I would really appreciate, Milan, that you can really, in an email engage with our attendees and answer their queries. These 2 questions, I'll certainly get back to you, and I would make sure that your response is actually taken to the people who has posted this question. So with this, I thank you all for your participation, and thank you for Milan for your time. Thanks everyone, and keep your support to us. Thank you very much.
Thank you all.
Thank you. Bye-bye.